Now, in many cases, we don't just have two alleles. We still keep talking about so many traits where it's just this or that. Normal blood cells, sickle blood cells. Healthy cystic fibrosis. We might talk about tall, short, purple, white flowers, whatever. In many cases, there'll be more than two. And so we're going to use the ABO blood type to talk about just the ways in which we can have multiple alleles. So for your blood type, and specifically we say ABO because there's other conditions that are part of your blood type beyond this. You have an RH factor, that's the plus or minus. There's other things we've discovered that we don't talk about as much normally. So we're going to keep it simple. For your ABO blood type gene, you can get the A allele, you can get the B allele, or you can get the O allele. Now, these are oftentimes written because you'll notice A and B are codominant. They'll oftentimes write them as IA, IB, and lowercase I. That way it really shows that A and B are both codominant. The I stands for immunoglobulin. That's just the particular chemical, so to speak, that's stuck on the blood that's causing this. And so you can have this A immunoglobulin stuck to your blood cells, your red blood cells. You can have this B one stuck to them, or you can have neither of them. So I is basically not producing any of these immunoglobulins. It's the lack of it. And it's a recessive trait not to have them. So we usually use the lowercase I to indicate this is recessive to both of these other ones. So now we can look at individuals to figure out what they be. So if you have two A alleles, you're going to obviously have blood type A. If you have an A allele and an O, you're going to be blood type A. Same thing if you have two Bs or a B and an O, both of those would phenotypically have blood type B. To have blood type O, you have to have little I, little I. That's the only way possible. So what we've got that's demonstrated over here is if we have one individual who has the genotype AO, and we have another individual, in this case the mother, who has the genotype BO. If we do this cross, if we kind of set up this Punnett square, what you're going to see is we can get the blood type AB. Notice because they're codominant, they both would be expressed. So this individual here is going to have blood type AB. So that'll be one of the people. Then you've got the next individual is going to get a B and an O, which would be ultimately phenotypically blood type B. So that would be another one individual. We then have this person down here that's going to have AO, which is going to be blood type A. It's one individual. And then down here we have an individual that gets two O alleles, and so they would have blood type O. Now this is kind of the most special case that I can get to. This one gives you the most possible blood types, but it's kind of cool that you could have a parent that's type A, a parent that's type B, and they could have every possible blood type, at least for the ABO group of children. Now, if you have a parent that's IAIA, or in other words AA, and you have a parent that's O, which if they have type O blood, they must be OO, you can only have one type. They're going to get an A allele from one parent, an O from the other, and then you know that they're going to be type A blood. So for me, I know my father's type A blood, I know my mother's type O, and I know I'm type A. So I immediately know my genotype. This is kind of like the pedigrees we did, where I know I got an O allele from my mom, I know I got an A allele from my dad, so I know that is my genotype, and I know my blood type is type A. If you want to get fancy and throw in the RH factor, I'm A+, obviously. So as you look through this, it's kind of cool that you can start to get more combinations now because you have three alleles and you throw in concepts like codominance. So I want you to realize that you can mix and match things where we don't just have multiple alleles, we also have where there's partial codominance, partial complete dominance. Genetics can get really interesting. That's also what can make it very convoluted as we try and study it, and that's why it was a great thing that Mendel started off with simple traits with just two alleles that happen to have complete dominance, which allowed us to kind of figure out the basic stuff going on so we could elaborate and start to understand stuff like blood typing as we know it. So if I give you problems like this, be prepared to solve them. Now the last bit in 11.2 is epistasis. So we're going to take this kind of odd genetic stuff and ramp it up a notch. So in this case, we're going to look at Labrador retrievers, although this can occur with a lot of things that are kind of like albinism. You can see epistasis occur a lot. So the way this works is labs have a particular gene, and for that gene they can have the recessive trait, which is chocolate, so they're producing a particular dark pigment in particular concentrations, and then they can also have the dominant trait where they have at least one large B, which is going to code for black, and so they would be a black lab versus a chocolate lab. So these are the two natural pigment states of Labrador retrievers. Now there is a separate gene, an epistatic gene, and what this does is it affects the expression, it affects whether or not we use another gene. And so in this case, if you have the recessive for that epistatic gene, so if we have two little e's for that one, it shuts off the production of that dark pigment, and so you get a yellow lab. So even though this lab has the particular alleles that code for being a black or chocolate lab, it won't be a black or a chocolate lab because those pigments will be shut off by a separate location, a separate gene. So if your parents put you in a room and you just want to go play video games, so you're like, all right, fine, I'll go to my room and I'll play them. You can turn on the video game console, you can turn on the TV, but I could in a sense epistatically control it by just going to the circuit breaker, flipping the breaker for your room, and say, have at it, see how that goes. And you ultimately will not get power, and it doesn't matter if all your stuff is in the on position, it's not going to work. That's effectively the way that epistasis works.